Imagine sending your best soldiers into battle. You arm them, train them, and send them after the enemy. But halfway through, you notice something strange. They start getting tired. Their aim wavers. Their coordination fades. Not because they are weak, but because their own bodies are telling them to stand down. That's exactly what happens to CAR T cells, one of the most promising cancer therapies of our lifetime, inside tumor-filled bodies. And now, a team of researchers from Columbia University and University Hospital Tübingen has discovered what's flipping the switch. The answer, published June 2 in Cancer Discovery, comes down to a single protein called NFIL3. Disable it, and CAR T cells stay in the fight much longer. Before we get to NFIL3, let's build the foundation. CAR T cell therapy is a form of personalized immunotherapy that works like this. 1. Collect. Doctors draw blood from a patient and harvest their T cells, the immune system's assassins. 2. Engineer. In a lab, those T cells are genetically modified to recognize a specific protein on the patient's cancer cells. Think of it as giving soldiers a photograph of the enemy. 3. Expand. The modified cells are multiplied in the lab until you have billions ready to deploy. 4. Infuse. The CAR T cells go back into the patient, where they hunt down and destroy cancer cells bearing that protein marker. The results have been nothing short of miraculous in certain blood cancers. Some patients with advanced leukemia, who had exhausted every other option, went into complete remission after CAR T cell therapy. But here's the catch: it doesn't work nearly as well for solid tumors like breast, lung, or pancreatic cancer. The tumors that make up the majority of cancers resist the attack. Why? CAR T cells don't just lose their edge because they run out of enemies. They lose it because the tumor microenvironment literally exhausts them, a process researchers call T cell exhaustion. Over time, the cells stop multiplying efficiently. They produce fewer kill signals. They become, in immunology speak, dysfunctional, and the tumor wins by attrition. The Columbia Tübingen team, led by pioneer Prof. Mitchell Sadelain and Prof. Judith Fucht, wanted to understand what molecular mechanism was driving this burnout. So they did something bold. They screened roughly 400 transcription factors, the proteins that control which genes get turned on or off, to see which one was most strongly associated with CAR T cell exhaustion. NFIL3 is a transcription factor that, when active, essentially tells the T cell to wind down. The researchers used CRISPR-Cas9, the famous gene editing tool often called genetic scissors, to disable the gene responsible for producing NFIL3. In mouse models across multiple cancer types, CAR T cells without NFIL3 stayed active longer, they multiplied more efficiently, they maintained stronger anti-tumor effects. Mice lived significantly longer. Switching off NFIL3 could be a decisive step towards significantly improving the long-term potency of CAR T cells, explained Prof. Fucht. This is important because solid tumors are the holy grail of immunotherapy, the area where CAR T cells have consistently underperformed. If removing NFIL3 helps CAR T cells persist in the hostile tumor microenvironment, it could unlock this therapy for millions of patients who currently have no options. That said, this is still preclinical. The team notes that human trials are needed before this approach can reach patients, but the path forward is clearer now than ever. This story sits at the intersection of several powerful technologies converging. Immunotherapy, harnessing the body's own defenses, CRISPR gene editing, precise molecular surgery systems-level screening, testing hundreds of variables to find the one that matters. It's also a reminder that breakthroughs don't always come from building something new. Sometimes they come from removing the thing that was already there, holding everything back. Question 1. What single protein did researchers identify as a key driver of CAR T cell exhaustion? Answer: NFIL3, a transcription factor that tells T cells to wind down and reduces their anti-tumor effectiveness. Question 2. Which two institutions led the research published in Cancer Discovery in June 2026? Answer: Columbia University, led by Prof. Mitchell Sadelain, and University Hospital Tübingen, led by Prof. Judith Fucht. Question 3. Why is NFIL3 discovery particularly significant for solid tumors? Answer: CAR T-cell therapy already works well for some blood cancers, but struggles against solid tumors. NFIL3-driven exhaustion is believed to be one of the main reasons CAR T-cells fail in solid tumor environments. Removing it may unlock the therapy for the cancers that need it most. Sources: Science Daily, University Hospital Tübingen, June 2, 2026. Cancer Discovery, Columbia University.